Assalamualaikum warahmatullahi wabarakatuh. Hello everyone, my name is Amazida Nur Insani with the last name 109. I will explain about enzymes and coenzymes. The functioning of human body consists of various systems built up by organs. Organs are formed from various tissues composed of cells that are specific to that organ. Cells contain organelles, which are subcellular components. Each organelle has a different function, like nucleus, DNA and NAD plus synthesis, mitochondria, energy generation, fatty acid oxidation, and lysosomes, macromolecular discussion. And the meaning is each organelle has different metabolic processes. So, in living cells, there are a number of complex and complicated metabolic processes, but they are interconnected with each other. However, these complex metabolic processes are perfectly organized and integrated to fulfill their function as specific cells. The process of organization and integration can be carried out thanks to the presence of enzymes that catalyze specific stages of metabolic processes. Enzymes are protein catalysts for chemical reactions in biological systems. Catalysts accelerate certain chemical reactions. Enzymes affect reaction speed by lowering activation energy. So, enzymes' function are catalysts and regulators. Enzymes are biocatalysts, are substances that have a biological origin, which can accelerate chemical change. In most regulatory mechanisms, enzymes participate. Almost all enzymes are proteins, but there are also catalytically active nucleic acid, namely ribozymes. Catalytic work of an enzyme or enzyme activity is measured by determining the increase in reaction speed under predetermined conditions. Enzyme characteristics. The largest group of proteins found in living cells, about 95% of proteins, are active proteins that function as catalysts. Enzymes function as organic catalysts, biocatalysts, made by living cells, but their activity does not depend on the cell that makes it. So it can be used in vitro. Enzymes require other components for their activity, or cofactors. Active enzyme complete with all its components, named by holoenzymes. Coenzymes or cofactors are very strongly bound and even covalently bonded to the enzyme. Covalent bond with the enzymes named by proteastic group. Part consisting of protein only in an enzyme named by apoenzyme or apoprotein. And the function of coenzyme is temporary carrier of functional groups that play a role in an enzymatic reaction. Inorganic cofactors are ions whose role is to help bind groups on substrates, molecules, and electron transport, e.g. Mg++, U phosphorylase, Ca++, U coagulation, muscle contraction, Fe++, Cu++, Rx oxidative. Like other proteins, enzymes have a BM between 12,000 to 1 million. Some enzymes do not require other chemical molecules for their activity, and some require cofactors or coenzymes. Cofactors are inorganic ions that enzymes need to perform their function. Coenzymes are organic molecules complexes that enzymes need to perform their functions. Naming enzymes. Enzymes are named after their substrate with the suffix -ase. For example, the enzyme sucrase breaks down sucrose into glucose and fructose. The old names of enzymes are still used, such as renin, trypsin, and pepsin. Enzymes are classified based on the type of reaction and the reaction mechanism they catalyze. Naming of enzymes: non-systematic pepsin, trypsin, thiolin, thrombin, and the others. Systematic EIB enzymes are grouped into 6 main classes. Each group is further divided into subgroups. 1. Oxidareductase oxidation reduction reaction. 2. Transferase. 3. Hydrolyze. 4. Lyase. 5. Isomerase. And 6. Ligase or synthetase. Oxidareductase catalyzes the transfer of electrons from one molecule to another. NAD plus and NADH are often involved in this reaction. Transferase catalyze the transfer of groups or atoms from one molecule to another molecule. Hydrolyze breaking carbon bonds with other atoms by adding water. Lyase catalyzes the binding of two molecules by addition to a double bond, or conversely, the splitting of one molecule into two new molecules at the double bond. Isomerase catalyze the rebinding of components of a molecule or isomer formation. Ligase or synthetase catalyze the formation of a substance from two substances and the splitting of a third substance. Factors affecting enzyme action. 1. pH. Each enzyme has an optimum pH to work. Example, pepsin works at pH 2, amylase at pH 7. Second, temperature. Every 10 degrees Celsius increase in temperature up to 40 Celsius degree. The reaction speed increases by 2 times and the reaction is inhibited and stops at 60 Celsius degree. And the third is concentration of S and or E. With the catalyst test, the activation energy is lower, EAB, but the EAC net energy remains. Role of enzymes in biological reactions. Enzymes increase the speed of spontaneous reactions at psychological temperatures. Distribution of enzymes in the cells. Cells, eukaryotes, contains organelles such as nucleus, ER, mitochondria, ribosome, and the other. Certain metabolic processes usually take place in certain organelles, e.g., glycolysis and Krebs cycle, named by cytosol, oxidative phosphorylation, mitochondria, protein biosynthesis, ribosomes, regulation nucleus, protein degradation, lysosomes. So there is a distribution or compartmentalization of enzymes in the cell. Certain enzymes are found in certain places, organelles, depending on the function of metabolic processes in the cell. In addition, there are also enzymes that work extracellularly, e.g. digestive enzymes. The purpose of compartmentalization to keep the metabolic processes in the cell from becoming chaotic. There are certain enzymes produced by certain organ cells, for example, EPL by vascular wall endothelium, hepatically passed by hepatocytes, CPK by striated muscle, LDH by cardiomyocytes, HHA by nerve cell synapse. Michaelis complex. Binding of the substrate to the active center, active site, will cause destabilization, loosening of certain chemical bonds, of reactive bonds which will cause easy bond breaking. The presence of COOH and H2OH SH groups on the active center of the enzyme has this catalytic effect. For combination with substrates, enzymes have one or more active centers, and in these active centers usually have OH groups from serine, imidazole from histidine, sulfhydryl from cysteine, which plays a role in substrate binding in the proximity region. Enzymatic reaction catalyzed by an enzyme SE complex PE E enzyme as substrate substance that undergoes chemical change due to the activity of an enzyme. P or product substance formed in the enzymatic reaction as molecule is bound to E in a specific area at the active site or catalytic site. When the enzymatic reaction has reached equilibrium after the reaction is complete, the enzyme does not change its structure. Regulatory subunit serves for the regulation of enzyme activity, containing one allosteric site. A cavity shape placed on the regulatory subunit as a place for where the effector or modulator is attached. To effector or modulator can be H. Inhibitor inhibits the speed of the enzymatic reaction. B. Activator activates the enzymatic reaction. C. Non-effector does not affect the enzymatic reaction process, used to prevent enzyme bonding with inhibitors. Some models of E is interaction. 1. Lock and key analogy. Enzymes have specific site structures that co-co with the substrates, can explain the specificity of the enzyme but cannot explain the stability of the enzyme-phase transition. 2. Induced feed theory considers inflexibility of proteins so that the binding of a substrate to the enzyme causes the site to change its conformation so that it cooks with the substrate. Can PT explain the phase transition of ES complex? Kohlen space, a new state of feed after a conformational change of the enzyme protein molecule following contact with the substrates or induced feed. Phosphorine V and SH contain residues bound in catalyst. Binding of the substrate causes a change in the shape or conformation of the enzyme protein, which can place the group appropriately for substrate and enzyme binding. Medical aspects of enzymes. As a biocatalyst is a protein, regulate metabolic processes so that there is no cause or damage to cells. Maintaining homeostasis, the tendency to regulate and maintaining in a stable environment in the body. So if there is a disturbance in the production of enzymes in cells, homeostasis is disturbed. The body becomes sick. Conversely, if the jar or organ is damaged, the production of enzyme from the cell is disturbed. Homeostasis is disturbed. Conversely, if the jar damages enzyme production from the cells, so that jar is disturbed, homeostasis is disturbed. Disease occurs, so enzyme disturbed and disease. Enzymes are often used as diagnosis tools. Enzymes and disease. SGOT, heart infarction, SGPT, hepatitis, amylase lipase, pancreatitis, creatine kinase, muscle and cardiac infarction, acid phosphatase, CF, prostate, alkaline phosphatase, GG-bone. Use of enzymes in the clinic. 1. As a diagnostic aid. In this case, the enzymes is available in kit or crystal form to determine the levels of certain substance in the body. If from normal of suffering from TTT disease. Example, determination of glucose levels, determination of blood cholesterol levels, determination of blood TAG levels. To determine of the course of a disease, there is a relationship between blood enzyme levels and an extent of tissue damage. Recovery when the enzymatic curve returns to normal. Poor prognosis when the blood enzymatic curve increases. 3. Determine the response to therapy. Enzyme activity is seen after the administration of appropriate therapy and diet. Maybe that's all from me. Please apologize if there are mistakes. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.